Hello, my name is Curran Howland, and I will be doing a presentation on site adaptations in teleost and conthicthes due to light attenuation. Throughout the presentation, my topics will be properties of water, which I will be focusing on the bending of light, and then from the bending of light, there's three subcategories I'll be talking about: refraction, reflection, and scattering. Then I'll move on to light attenuation from the surface to and onwards down through depth of the ocean, and then color scale of how we lose the colors that we see normally as our at the surface are our reds, oranges, yellows, but then we start to lose our greens and then eventually only blues. Then I'll move on to the layers of the ocean. Our photic zone is our sunlit surface waters in which photosynthesis is able to happen, and aphotic zone is where it is completely dark. And then we talk about the site adaptations in teleost, which is the bony fishes, which can be your tunas, clownfish, and most predominantly all of your reef fishes. And then we'll compare those to the conthicthes, which is comprised of sharks, skates, and rays. And then the final thing I'll be talking about is site, and then the comparison of the strategies of both of these groups in the photic versus aphotic zones and how those site adaptations will help them, and then the inner workings of the eyes themselves, and then the connection from the neutral images that they perceive, and then how that transfers into the brain and how they understand it. As I stated before, I'll be talking about the bending of light, and this can be broken into three categories: reflection, refraction, and scattering, as I said before. Both reflection and refraction can be seen in the top right image, and then scattering can be seen on the bottom one. Reflection, I'll start with that one first, is a special case of scattering in which a large surface area redirects radiation in a backward direction. This can be seen at the top portion of when the incoming light hits the medium of air and water and then is reflected back up into the air, while refraction is when light penetrates from air into the water, thus slowing down the particles, causing it to change direction slightly. You can see this as the incoming light hits the water and then it slightly changes the angle of how the light is going into the water. And finally, scattering, which is the particles disperses radiation in all directions, forward, backward, and sideways. You can see this in the bottom portion when the light waves are coming from the left, hitting all the tiny little particles and then scattering in all different directions. These are the sources. Alright, properties of the ocean water. What light attenuation is. What is the main, why am I talking about this? So this is the gradual decrease in light intensity as it travels through matter. I will be focusing on water, the ocean water. The color scale and wavelength. This image on the right shows light penetration underwater. On the left, it is the open ocean under very clear conditions, versus on the right, which is the coastal or clear freshwater. But I'll be focusing predominantly only on the left side. So the full spectrum of light fades out around 30 meters of depth. Remember to stay on the left side of the image. And this is, you have all your reds, yellows, and greens, blues, purples, and violets. And then green stops out around 175 meters. And then blue light penetrates to about a depth of 200 meters. And then as all these numbers and ranges can be changed due to turbidity. And turbidity is the amount of stuff that's in the water this can be due to waves and plankton blooms and other forms of, and sediments. And there's that. And this video will show of how this really does work in the actual open water with a diver and an apple.
red because molecules in its skin interact with the various wavelengths of sunlight. Now, most of these wavelengths are actually absorbed by the apple, except red, which is reflected. Now, our eyes detect the reflected red light, and our brains register that we're seeing a red apple. It seems pretty simple, huh? Well, it's not so easy to explain the colors of many things, like water. So we're going to explore the question, why is the ocean blue? And to do that, we're going to run a simple experiment. All we need is this red apple. We're going to use these colored toppers, and we're going to use my dive gear. Because you see, I'm going to dive down into the depths, and I'm going to see if the colors look any different underwater. Well, what do you think's going to happen? I don't know. Let's go diving. Here we are at 1 meter, and the apple still looks red, right? Well, now let's descend to the wreck at about 35 meters. The apple looks dark gray or black. Did the apple really change color? No. When I turn the camera light on, the apple is still red. And look, we can see some of the colors here on the toppers. Do you know why we can't see red, but we can still see green and blue? Well, at the surface, the apple looks red because it reflects red wavelengths. But at this depth, there doesn't appear to be any red wavelengths for the apple to reflect, and it absorbs all the other wavelengths, so it looks black. But what about the color toppers? You can see the blue and green colors are still showing up, even if they appear a little bit different. So that means that at this depth, there might not be any red wavelengths, but there's still some blue and green wavelengths. So why does this happen? It's because red wavelengths are absorbed by the water first, and blue and green wavelengths penetrate deeper into the water. All right.
Okay. That was a quick video, and it's an easy way to understand. He was only 35 meters down, and that is truly only about 100 feet. But you'd still need to go down to 200 meters to actually leave the photic zone, which I'll be talking about next, about within the layers of the ocean. Okay. And then this is on the image you will see the photic zone of the first 200 meters, and then this is the sunlit surface layer of the ocean down to a depth where light is just sufficient for photosynthesis. And then meanwhile, in the aphotic zone, there is part of the ocean where light does not penetrate and no photosynthesis occurs. There are two portions within the aphotic zone. One is called the twilight, which you can see is between the photic zone and the no light portion on the right. And this is an area that there is little downwelling incoming light, and organisms are adapted can still see. And then the second portion is that range. This region can be ranged from 200 meters to either 1,000 or 4,000 depending on locations. When you get to the second part of it is where there is no light penetration, it is completely dark, and this is when it is getting closer to the ocean bottom. This depth can be ranged from the twilight zone to either 6,000 to 10,000 meters to the ocean floor. Photic zone. So this consists of the euphotic zone and the dysphotic zone. The euphotic zone is the upper part of the ocean that receives bright and clear light, sunlight. The dysphotic zone, the water layer beneath the euphotic zone, where dim blue light penetrates but is not sufficient enough to sustain photosynthesis. Organisms use sight to find their food with available light, such as like sharks and and tunas and stuff like that. They all look for visual cues within the water to find their prey or evade. And this occurs again at the ocean surface to about 200 meters deep. And then the optimal wavelength absorbance is 500 to 530 nanometers. This is like your reds and your oranges and all that. The aphotic zone, this is the portion, again, as I stated before, where there is no visible light at all. Organisms use different sensory skills instead of their eyes, such as chemosensory or they have specialized eyes to help them see in low to no light at all. This ranges between 2 to 1,000 meters depth or to the ocean floor. And then wavelength of light absorbed best is 470 to 480 nanometers. And this is your blues and maybe your greens, but not very much. The types of organisms there. So the teleost and then the Canthrichthyes. The first two images that popped up are in the photic zone. So you have on the left your yellowfin tuna, and then for the Canthrichthyes you have the great hammerhead shark. And then for in the aphotic zone we have for the teleost a barrel eye, and then the Canthrichthyes the Portuguese dogfish in the aphotic zone. Each has been adapted over time to see and be able to hunt and evade other predators. Sight. So on the right you have a representation of the teleost eye, and then you have the three main functions. So it is collecting light, focusing images on the retina, and then transforming images into neutral signals. And if the organism does not know the individual image, they can use clues from the surrounding area to relate it to something they have encountered beforehand. I will further go into individual aspects of the most important parts of the eye, and it will pertain to both teleost and Canthrichthyes as they are only varying in slightly. Okay. The eye is composed of 7 basic parts, as there are more, but these are the critical for sight. I will individually explain the importance of each as they show up on the screen. As you see, we still have the same diagram as before, but we actually have some parts that are labeled. So the cornea, which is right there, is which acts as the eye's outermost lens. Then the iris is right there, and it controls the amount of light entering the eye by adjusting the pupil, which is the black portion of your eye that you see that you actually have in your own eyes. The lens, which focuses light on the retina. The sclera, right there, which forms the outer layer of the eye to protect the inner structure. The choroid and the tapetum lucidium, right there, which sometimes work together to reflect light through the retina back into the inner eye of the retina, so that it increases the light efficiency and photoreceptors can be able to detect the incoming signals. And then the retina itself, which this can be seen in many organisms as the characterized the reflective shine of the eye in the contact. That was for the tapetum lucidium, and then, sorry I got that mixed up, but finally the retina. The purpose is to convert the reflected light into neutral signals and then send it to the brain. Teleost. So this group is very diverse species, as it ranges from our fast-swimming fishes, both yellowfin tunas and the swordfish, to the clownfish that it calls its reef. So then we have the aphotic zone. This is your barrel-eyed hatchetfish and anglerfish. The diversity is large, thus relating to the different types of eyes you can see on the picture to the right, along with the photic fishes and the aphotic zone, a large population. But no matter where the fish is located, the size grows throughout the entirety of the fish's life. And then you see again the retinal tapetum, which is a retroreflector, reflecting visible light back through the retina, increasing available light to the photoreceptors. This is critical for the dysphotic zone and then the aphotic zone, as there's barely any light coming down, penetrating through the ocean, thus downwelling into the air where they live. So the teleost sight strategies. Twilight to aphotic. So predominantly beforehand, in the aphotic zone, they just use their eyes just to see everything. But these ones are special as they have adapted to living in their area. So they have the yellow intraocular filters. This function is the reduction of chromatic aberration. And chromatic aberration is the material effect produced by the refraction of different wavelengths of electromagnetic radiation through slightly different angles, which results in a failure to focus. And then a reduction of glare, thus improvement in the contrast vision. So if they are looking up, it makes it a little bit easier to see the outline of either their prey or their predators. A transparent head. The green eyes filter out sunlight coming from the ocean above. This is especially helpful if you're in the dysphotic zone with still downwelling light, and then also if there's bioluminescence, you can see everything a little bit better. And then your tubular eyes. Large lens, which is good for lighting properties and looks predominantly upwards. And then this video. The video that I'm going to show puts two of the three strategies together in one organism, the barrel eye.
It is a small, dark fish with large eyes, a tiny mouth, and a remarkable pair of eyes. The two green spheres in these video shots are the lenses of its tubular eyes. Behind the eyes is a set of large scales. The eyes are enclosed within a transparent shield, sort of like the canopy of a jet fighter. In front of the eyes are two dark capsules containing the fish's olfactory organs. Typically, Macropinna sits quietly in the water, using its big fins for stability, while it scans the water above for food. When it spots food, it can rotate its eyes to look forward, to include its mouth in the field of view. We speculate that Macropinna steals food from siphonophores, elongate animals with tentacles that capture prey that swims into them. And we think that Macropinna swims into the tentacles and steals the food from the siphonophores. The shield over its eyes protects those sensitive structures from the stinging cells in the siphonophores' tentacles. It can also rotate its eyes to avoid predators, or to avoid being captured by scientists. This is Bruce.
And again, that was the barrel eye, which lives in the aphotic zone. So the next topic will be the components of the Conthicthes eyes. So their main difference from teleost is that they have a nictitating eyelid. You can see this is on the figure to the right, the moving actual part that's going to cover the eye. This protects the eye during an attack. Very famous organism that does this is a great white shark. As it attacks a seal, it covers its eyes to protect it so that the organism cannot scratch or hurt the actual shark. And this video will show a more in-depth way of how this organism uses this, plus the tapetum lucidum to help it see in the dark.
Shark has a reflective layer behind the retina called the tapetum lucidum. Its purpose is to reflect light back through the retina a second time. It's very similar to the eye of a cat. If you look into a shark's eye at night with a light, it'll actually reflect back at you, just like a cat's eye. This increases the low light sensitivity of a shark's eye. They can see amazingly well in light that to us would just look dark. Some estimates say that the shark's eye is 10 times more sensitive in low light than human eyes. And get this, sharks also have eyelids. They're the only fish with eyelids. They're called nictitating membranes, and they close them when they're attacking prey at close range to protect their eyes. Sometimes
The next topic we'll be talking about is the Conthicthes vertical distribution, as they aren't as predominant as teleosts in population size, but they are still found throughout the different layers of the ocean. So on the right you see our distribution of major known sharks. As you see, most of them are within the sunlit euphotic zone at the top, or the photic zone, as I was saying before, then into the twilight zone. Very few of them actually live in the aphotic zone, but in the photic zone, we have the great hammerhead, the great white shark, and then the whale shark. While the aphotic zone you see at the bottom is the Portuguese dogfish and the frilled shark. And Conthrichthyes sight strategies. Like in the ocean, like the teleos, the eyes continue to grow throughout the lifespan of the Conthrichthyes itself. Um, the pelagic and mesopelagic, they have the largest eyes to allow for available light penetration. As they are both visual and top predators, they need to be able to see the most amount of incoming light and images. The bathypelagic have smaller eyes but with larger pupils. This is to increase the visual acuity and ability to detect bioluminescence. As I stated before, how how teleos can also use um, their pupils to let the incoming light and also decrease the amount of incoming light. So can the Conthrichthyes. And then the aphotic zone, the large eyes, immobile pupillary, so they can't move that, and they have a low focal ratio, which equates to an increased detection and amplification in low light. So if they can't move their eyes, it's very set, and they have a set distance of what they can see, but they can see very well. And then the video I'm about to show you just shows what the actual sharks in the aphotic zone use to actually detect their prey.
Sharks that live in the deep ocean have evolved to have special eyes that help them see in the low light conditions. Researchers have discovered the science behind the bioluminescent shark's eyes by comparing them with the eyes of other sharks. Their eyes allow them to see the points of light that reach the depths of the ocean and recognize other bioluminescent fish. One important discovery made by the study found that there is a space between the lens and the iris of the shark's eye that lets extra light into the retina. According to study researcher Julien Claes, a biologist at the Catholic University of Louvain in Belgium, there are about 50 different shark species that are able to produce light, about 10% of all currently known sharks. They live at depths of between 650 to 3,300 feet below the surface of the ocean in an area called the mesopelagic twilight zone. Being able to produce light helps to keep them hidden from predators by camouflaging against the sunlight above. It also helps the sharks identify potential mates and hunting partners that belong to the same species.
And then we'll move on to the next one. So this is the similarities between teleosts and Conthrichthyes. So both of these two groups are able to warm the areas around the eyes to increase the retinal rate of response to the change. This is also known as the retiniere, which you can see is on the bottom right with the red line and then the blue line right behind the eye. This is called a counter-current heat exchange, which the red is warm blood and then the blue is cold. And then I will show you a video that actually really does explain how this works, and an in-depth version of it's a very simulated, but it shows a very good portion of why this works.
In order to maintain optimal some animals go through a mechanism known as counter-current heat exchange. This mechanism allows for the animal to save energy by using parallel pipes, which are the arteries and the veins, that allow blood to flow in opposite directions to and from the appendages. Here the arteries are displayed in red, and they are typically found deep within the appendage, while the veins, displayed in blue, are superficial. They connect in the middle by capillaries. For the purpose of display, the pipes seen in the outer part of the model exemplify regular blood flow which does not conserve heat. Rather, heat would be lost to the environment, thus steadily declining blood temperature. This process is held when the organism is at optimal warm body.
And since you got to see that good demonstration of how that countercurrent exchange works to help increase the visual, the warmth behind the eyes, which then allows them to perceive better incoming information, we can move on to the sight eyes perception. Sorry. So on the right, you see an image, a figure of the purple is a rod, and then the green is a cone. These occur in the retina, which allows for uh, photoreception pigment. And then the rods and cones. There, the outer segment contains a visual or the photosensitive pigment, held in place by an external limiting membrane. So individually, the rod, as in the purple, responds to dim light but with poor sharpness and without color perception. So this doesn't detect the greens and blues and reds, it's just black and white. And then this contains more visual pigments and is more sensitive. While the cones, the green image, is divided into populations, and each population has a different visual pigment. And when I forgot to mention that what a photosensitive pigment is. It are unstable pigments that undergo a chemical change when they absorb light. Okay, on to the next part. So the sight adaptations to change in light. So on the right you see the light and dark adaptations in teleost. On the left portion you see a dark adapted, and then on the right you see the light adapted. And at the top portion it is the direction of incoming light, and the black line that says external limiting membrane is what keeps the rods and cones in place. So we have the light. So when light happens, rods are farther from the external limiting membrane and farther from the light source. As this is on the right, you don't want your rods to be too far close because there's a lot of light incoming. And while the cones are closer to the external limiting membrane and then thus are closer to the incoming light. While the dark on the left is rods are closer to the external limiting membrane and closer to the light source. So you would want this because you want the most amount of incoming light to hit the rod so that you can perceive information. While the cones are farther from the light source and external limiting membrane. Okay. The connection to the brain. So you see that on the right, the image shows an actual full out from the retinas, rods and cones to the ganglion cells and the optic nerve. So the rods and cones are connected to the brain via bipolar and ganglion cells. This is normally on the subject, the figure part B. This is in between the rod and then the ganglion cells. The axons of the cells pass along the optic nerve to the optic lobe. And then all vertebrates use similar structures of the eye, including fish, constrictors, and humans. So I don't have individual images of them because it's all pretty much the same. And then I'm going to show you a video that explains this in further detail and how it actually happens.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the retina. The retina contains the neural component of the eye. When light reaches the back of the eye, it enters the cellular layers of the retina. The cells of the retina that detect and respond to light, known as photoreceptors, are located at the very back of the retina. There are two types of photoreceptors, rods and cones. Rods allow us to see in dim light, but don't allow for the perception of color. Cones, on the other hand, allow us to perceive color under normal lighting conditions. Throughout most of the retina, rods outnumber cones. In one area called the fovea, however, there are no rods but many cones. The fovea represents the area of our retina that provides our highest acuity vision, and thus is at the center of our gaze. When light hits photoreceptors, it interacts with a molecule called photopigment, which begins a chain of events that serves to propagate the visual signal. The signal is transmitted to cells called bipolar cells, which connect photoreceptors to ganglion cells. Bipolar cells pass the signal on to ganglion cells, which leave the eye in a large cluster at an area called the optic disc. The optic disc doesn't contain any photoreceptors, and so represents an area of the retina that can't process visual information, creating a natural blind spot. However, we normally don't notice our blind spot. The brain uses information from surrounding photoreceptors and the other eye to fill in the gaps in images that are processed by the retina. After leaving the retina, the ganglion cell fibers are called the optic nerve. The optic nerve carries visual information toward the brain to be processed.
And with end of that video, we get to the end of this presentation. So in conclusion, light attenuation determines the depth of the photic zone itself in the ocean. Thus, it is the decreasing available light constricts organisms to adapt to the little or no light environment. The aphotic zone starts at a depth of approximately 200 meters, depending on the location. Sight has three main functions in either teleost or the conjunctives. First is collecting light. Secondly, it is focusing images on the retina. And third and finally, transforming images into neutral signals. And then between both of the groups, the size of the eye is dependent on the layer where the organism inhabits. And then retinas are made up of both rods and cones, just to reiterate that, this is very important. And then the connection to the brain. So we start from the incoming light hits into the eye, comes into the eye, it hits the back of the eye, which is reflected off the lucidium tapetum, and then is bounced back into the retina, which then hits the rods and cones, and then the photoreceptors, bipolar cells, ganglion cells, optic disc, and optic nerve, and then finally the brain. And these are my citations and sources. Thank you. Have a good day. Thank you for watching.